Hello everyone, my name is Hanegraef and I'm based at the Natural History Museum in London. The research I'm presenting here is joint work with Fritz Bohr. In this presentation I will be talking about the maxillary morphology in African ape subspecies and the implications for human evolutionary research. The number of species recognised in the human evolutionary tree has increased considerably in the last 30 years, including hominins from the late Miocene, Middle Pliocene and Pleistocene, as well as dwarfed island hominins. But debate remains whether all of these deserve species status or should be included within the variation of existing species. Morphological differences among great apes are frequently used in attempts to recognize species in the human fossil record. Yet it is important to have a good understanding of the degree and structure of intraspecific variation, including aspects such as sexual dimorphism and geographical diversity. Subspecies of African apes were first recognized as geographical clusters, but are increasingly well defined based on genetic grounds. It was a mitochondrial DNA study that recently suggested the existence of a fourth chimpanzee subspecies, Pantroglodytes ellioti. Despite this genetic evidence, it is less clear whether the African ape subspecies show consistent morphological differences. Over the years, many studies have looked at the morphological differences in the cranial and dental morphology of African ape subspecies, yet most are based on qualitative observations or linear measurements. Shape analyses through geometric morphometrics give a more complete picture, yet previously only a subsample of the African ape taxonomy or relatively small sample size have been used in these studies. The sample analysed here is, to our knowledge, the most comprehensive thus far and based on current consensus views of hominid taxonomy, including the commonly accepted subspecies of Pantroglodytes, gorilla and gorilla beringei. We compared our results at the genus and species level by including 61 Pampaniscus and 106 Homo sapiens specimens. We explored the differences in the maxillary morphology of these African ape subspecies. Maxilla are the most centrally located bones on the face, structurally linking the orbits, nasal cavity and zygomatic arches, as well as the mandible via the dental arcades. Furthermore, maxilla feature in many hominin species descriptions, such as Paranthropus. This research is based on CT images, and the shape of the maxillae was captured using 68 three-dimensional landmarks, capturing the dental arcade shape, the spatial arrangement of the teeth, and the dental size proportions, as well as the heart palate, the subnasal morphology, the nasal aperture, the orbits, and the anterior-inferior takeoff of the zygomatic process. The maxillary morphology is able to differentiate between the Pantroglodytes subspecies, although with a substantial degree of overlap in shape space, as shown on this PCA plot. Maxilla of Pantroglodytes virus are the most distinctive of three subspecies, and as shown on this slide, they are relatively wider, lower, and shorter than the other two subspecies. Maxilla of Pantroglodytes troglodytes and Pantroglodytes schweinfurti are more similar to each other, yet they do differ in certain aspects of their morphology, with Pantroglodytes schweinfurti maxilla relatively narrower, more prognatic, and longer than those of Pantroglodytes troglodytes. The maxillary morphology is also able to differentiate between the gorilla subspecies, yet again with a substantial degree of overlap in shape space. Maxilla of gorilla dieli, pictured here, are relatively narrower, lower, and longer than those of gorilla. Additionally, the subnasal segment is more prognatic in gorilla dieli. Gorilla beringei subspecies can also be differentiated based on their maxillary morphology, again with a substantial degree of overlap. Overall, the maxilla of gorilla beringei groweri are relatively wider and slightly higher than those of gorilla beringei. The anterior maxilla are relatively shorter in gorilla beringei groweri, but its posterior part is relatively longer. Looking at the amount of morphological variation in the context of intergeneric, interspecific, and intraspecific variation, the gorilla subspecies are morphologically more diverse than even Pantroglodytes and Pampaniscus, and the gorilla beringei subspecies even differ almost as much from each other as do gorilla and gorilla beringei. The maxillary shape differences among the subspecies of Pantroglodytes are less than that observed in the subspecies within gorilla. The relative lack of differentiation among the Pantroglodytes subspecies found here is quite interesting from an evolutionary point of view, and several theories have been put forward. First, it could be possible that the lower morphological variation reflects a more recent differentiation of the subspecies of Pantroglodytes than those of gorilla. However, a 2013 study characterizing the genetic variation in great ape subspecies shows that the split times, which are the dark brown lines on this figure, are earlier between the chimpanzee subspecies than for the gorilla subspecies. Another possibility is that chimpanzees have simply maintained higher levels of contact among their populations, thus increasing gene flow and inhibiting differentiation. In addition to having a continuous distribution with no major geographical barriers, chimpanzees are extremely mobile animals with large home ranges and an unusual social organization known as fission fusion. A third possibility is related to ecological factors. Chimpanzees utilize a range of habitats, and this does not appear to correlate with the major geographical groups of Pantroglodytes. In contrast, gorilla subspecies inhabit somewhat different and more specialized ecological zones in terms of altitude and dietary preferences. The results of this study confirm that the African ape subspecies are not only geographically and genetically defined, but can indeed be characterized using aspects of facial morphology, in particular the maxilla. Moreover, they demonstrate that the maxilla can provide useful information when exploring taxonomic diversity among hominids at a genus, species, and subspecies level. Exploring the degree and pattern of morphological variation of the maxilla in and between African ape species and subspecies will contribute to a better understanding of the hominin fossil record, in particular in relation to little-known species such as Canyanthropus platyops, Australopithecus dearomeda, and Homo rudolfensis. In conclusion, and answering the question on the first slide, yes, subspecies of African apes can be distinguished based on their maxillary morphology, although there is substantial overlap in shape space. I would like to thank all the museums, institutions and curators that provided access to their CT scans, and thank you for listening.